And then, all right, welcome to another week of Soul Seminar. Today we're actually going to start off with a little announcement from Dr. Sandy Knapp. So you can start whenever you want.
Okay, can I share my screen? There we go. Okay, I just want to announce to people that the Solanaceae genomics community, which usually meets every year in a place all over the world, had canceled the meeting for this year, which was going to be in Greece. And it was decided by the kind of organizing committee of that meeting to hold an international online meeting that would be free to register. And I just want to show people how to register, because this is something anybody can come to. It's open, it's free, and we are giving preference to people to give talks and posters to people who normally would not come to these meetings because of financial, for early career researchers, or people who couldn't come because it's way too expensive to go to these meetings every year. So what to do to register is you go onto the Solanaceae source web page, top front page, and then the first item under Solanaceae News is a little description of the meeting. And if you click on this Sol International Online link, if this all works properly, it should take you to the landing page, which is on the Sol Genomics Network, which is where you register, and you can submit an abstract in the same form, and your abstract will automatically go to the person who's registering, who's overseeing that section. So you just fill in your email. So I'll just do it again, and you can see what happens. And then there's registration, and then it asks you if you want to do an abstract, and it just takes you on, and you do the abstract. And then if you want to speak in the biodiversity section or have a poster in the biodiversity section, the thing will automatically come to me. So please do register, especially early career researchers and people who normally don't come to these meetings, because I think it's important for the genomics community, the people who are working on crops, to actually see the amazing diversity that's in the family and see the kind of scope of what it is that people that work in Solanaceae do. So thanks very much, Chelsea, and everybody register.
Awesome. Thank you, Sandy. Sounds like a good conference to be a part of for sure. So today I'm going to introduce Boris Igić. Boris grew up in Bosnia, where he mostly played basketball and other street sports, but after moving to the United States, he had more of a scholarly view. Got his undergraduate degree at Washington University in St. Louis, and then continued on with his research at the University of California in San Diego. He did a small postdoc at Cornell University, and he is now the professor at the University of Illinois in Chicago. Boris's work mostly focuses on plant mating and breeding systems, but his lab has also done work in models of trait evolution, species delimitation, pollination ecology, and bird phylogenetics. Today, Boris will be talking about self-incompatibility in Solanaceae and beyond. And with that, you can start your presentation, Boris.
Well, it'll take me a second here. All right. Thank you so much. I really appreciate this. First, I would like to thank Chelsea, Andres, and Rocio for organizing these wonderful opportunities. I just think that's a great idea, and I'm so happy that you guys started these even before COVID. I'm flattered to be invited. Thank you very much. Before I begin, I'd first like to just say a few words about the family. And I don't mean the plants, I mean the Solanaceae family of people to whom I have this sort of great privilege to address. The nature of evolutionary research projects in my lab is such that we often rely on your hard work and generously shared findings. And in our work on breeding systems, we stand firmly on the shoulders of Greg Anderson and Lynn Bowes and Mary Kellen Arroyo and Sandy Knapp and Dick Olmsted and David Spooner and many others, including the person who inspired much of my work, Michael Whalen. He died when I was a 9-year-old boy in Yugoslavia, and I've obviously never met him, but his ideas are remarkably influential, and he would have turned 70 years old earlier this year. And so we drink to his memory every May in my lab, and I just wish for his memory to live on. With that in mind, I'm happy to present our collaborative and codependent work to the community that generated so much of the data that we critically depend on. In return, I hope I can inspire you guys to feel a fraction of excitement and joy that we feel about both past and ongoing research on trait evolution, macroevolution, and breeding systems in Solanaceae and other families, as well as the implication and expansion of this work beyond the family. So with that, I'd like to begin the show. So here's the outline of today's talk. What will happen first is I will briefly take some time to introduce self-incompatibility and show you some previous work. We'll talk about the definition of SI and the distribution. We'll go through some basic principles and genetic causes of SI, especially in a particular system that's often called the Solanaceae system, much to our delight. We'll discuss the consequences of self-incompatibility, perhaps why SI loss rates are so high, whether some self-compatible species, and how they may be balanced, these losses may be balanced by a higher diversification rate. There are also some challenges in inference and macroevolutionary inference that I'll address very briefly, and then move on to how we can use all this data to make some inferences as well as expand the known SI universe to Cactaceae and maybe examine some of these macroevolutionary inferences. All right, so if you're watching this, you already agree that the seduction of flowering plants is mesmerizing, both visually and by the numbers. There are about 300,000 species of angiosperms, and they're at least 140 million years old. Of course, the timing game is a little complex, but anyway, they show this bewildering diversity of morphological, physiological, and life history traits. But the real showboater among the traits of angiosperms is the mating system variability. So the variability in sexual breeding systems and mating systems is a real hallmark of this group. And mating patterns have a pervasive effect on organismal evolution because they affect the amount and distribution of genetic diversity in populations, and in this way they affect the evolution of all other traits. Now, of course, flowering plants are sessile. Oh, by the way, here's our, in case you were, this is kind of a little, where's Waldo? And here's Waldo. So plants are of course sessile, and their ability to attract pollinators and choose mates is governed by flowers. And over 90% of flowering plants are hermaphrodites, and they're simultaneous hermaphrodites very often. And therefore they can at least potentially self-fertilize. But most do not, as you're well aware. They express traits such as hercogamy and diaecy, and perhaps the most common mechanism to ensure outcrossing, an invisible trait of great importance, self-incompatibility. So let's formally define self-incompatibility as the genetically enforced inability of simultaneous hermaphrodites to self-fertilize. So these are otherwise fertile hermaphrodites that cannot self-fertilize. And the flip side of this is self-compatibility, which is the absence of self-incompatibility. In other words, some plants do not recognize and reject their own pollen. And this is a widespread phenomenon. So the reason why I'm sort of bringing this up right now is because it's good to start with common terms, but also there's this immediate realization that perhaps there can be some interesting dynamic. If self-compatibility is merely the absence of incompatibility, you can imagine how whatever causes incompatibility, those genes can be broken down, incompatibility can be lost, and at some point it clearly had to be gained. And so there is a potential here for a dynamic. And that dynamic is really at the heart of what my lab has been doing for the last maybe 15, 20 years. So self-incompatibility is not a rare phenomenon. It's expressed by about 1/2 of all flowering plants from more than 120 families or 125 families. We're sort of starting to actually formally count this. Obviously, the family changes are a bit of a problem, so taxonomic changes are a problem, but so far it seems that it's about 130 families. There are about at least 35 unrelated SI systems that occur in different lineages. The inference of that number is a mere inference based on position and some assumptions, but we know in at least 4 different cases that there are these systems that are non-homologous. So, for example, in Brassicaceae, there's a system which is completely caused by different genes than in Papaver, which is caused by different genes, again, different genetic bases than in Primulaceae, which seems to be caused, all of these seem to be different than the system in Poaceae, which we're not certain about, but we have a pretty high confidence that it's completely independent of the other 3. And perhaps most interestingly, one system is widespread and it's present in distantly related families which seem to share a homologous basis of self-incompatibility. This is the Solanaceae system. It was first discovered in Solanaceae by, I think the first publication on this topic implicating these genes is by Anderson et al., by this large Australian group. Then McClure et al., and then a bunch of work both in Melbourne and then by David Cow's lab at Penn State, where they've done just a tremendous amount of work, really heroic effort to clone the system and describe its workings. And either identical or very, very similar systems are expressed in Plantaginaceae, so Antirrhinum and its relatives, in Rosaceae, in Rubiaceae, and this is a recent discovery in Rutaceae. So it's not just confined to one family, and I'll come back to this later, it'll become important. But for now, let's get back to this dynamic, right? So it's possible entirely that self-incompatibility could just be lost frequently, and that's the end of that, right? And in that case, we would not observe self-incompatibility presently, but there are things that maintain it in populations and species. So one thing that is sort of a barrier against loss is inbreeding depression, the expression of inbreeding depression. Another one are long-term trends, as I'll hope to convince you a little bit later, which is that, for example, things like decreased extinction and increased speciation could maintain the presence of self-incompatibility in flowering plants. In other possibilities, that there are things that favor self-compatibility. In other words, that these mutations that change incompatibility to compatibility are favored in some way. There could be trivial causes, such as, for example, there could be mutational bias. So that, for example, the incompatibility genes are a pretty big target and they can be very easily mutated, but that the reverse is not true. So that exact reversals are very difficult, and then re-evolution of the system could be, for example, difficult. We know that loss of self-incompatibility occurs frequently. So approximately the mutation rate is about 10 to the minus 7 to 10 to the minus 5, or 10 to the minus 5, 10 to the minus 7. And we also, this is, by the way, if you imagine per pollen grain, so per flower, there could be, in some species, a single mutant basically in every single plant, right? So then these new mutants can experience an advantage, an automatic advantage, if they are indeed able to self-fertilize. And then they could experience some advantages with mate limitation. In other words, when mate limitation is present in populations, the ability to reproduce uniparentally can induce a strong advantage. So in cases, for example, where pollinators are rare on oceanic islands and so on. And we have this kind of advantage sort of codified, and we've done some work on this in the lab and with collaborators. So I'm going to first show you one of the cases, or showcase one of the cases where perhaps self-compatibility is favored, and that would be in dispersal to oceanic islands. So this is commonly referred to as Baker's rule, and I'm going to define it as well. So Baker's rule, at least in my opinion and in the opinion of the co-authors on this paper, is the prediction that under a variety of circumstances, uniparentally reproducing species are superior colonizers. And this is nearly verbatim taken from Baker's paper. So under a variety of circumstances, you can imagine that, for example, mainland species can migrate to islands, or within metapopulations that occupied patches can, individuals can disperse to unoccupied patches, or that you can have something like center margin dynamics, so that continually expanding and contracting species during the expansions the compatible individuals might have some advantage, or uniparental individuals, uniparentally reproducing individuals might have some advantages. So what Baker's rule predicts is an association between colonization and breeding systems. So it's essentially a trade correlation that would arise. It explicitly considers the role of ecological dynamics, which is one of its sort of key attractors. And it's a simple and general model, and it has some wide potential for applied interest to invasive species and conservation. And so its key prediction in our context is that self-compatible taxa should be more successful colonists than self-incompatible ones. And so we set out to test this. So this is one of the nascent groups of which I was a part. What we decided to do is test the ability to colonize islands, whether remote or not. But before we did that, we spent a lot of time talking about and thinking about what exactly Baker's rule is referring to. And because the problem of colonization is general, and then the breeding systems are just a part of colonization story. So you can imagine, for example, that you have an original location, let's say a mainland, and that there can be some opportunity, some rate of long distance dispersal. So that dispersal ability itself will affect colonization success. But what we're interested under Baker's rule is just the filter that breeding system or ability to uniparentally reproduce represents, so the barrier that it represents to colonization, or the filter of colonization that it presents. So that if you have, for example, self-incompatible and self-compatible propagules arriving on these islands, what you can imagine is that self-compatible taxa in absence of pollinators may have some kind of an advantage during establishment. And then also what Baker's rule ought not to include is subsequent evolution of breeding systems. So if these propagules establish already, and then subsequently they evolve self-incompatibility or other forms of outcrossing, that really should be inadmissible in its relation to Baker's rule. And so what we did is we set out to test this, and specifically the way we set out to test it is to sort of look for this weak signal. What we expected would be a weak signal of caused by the predictions that the Baker set forth, and then realized by the different frequency of self-incompatibility or compatibility in their occurrence on either mainland or island geographic groups. And so if Baker's either wrong or the signal is incredibly weak, what we expect is that the frequency of self-compatibility and incompatibility on mainland and island habitats would be approximately equal, or anyway not significantly different. Or, for example, it could be that self-incompatibility is even more common on islands, counterintuitively. If Baker is right, what we could, for example, do is estimate the advantage, the proportion sort of variance that can be explained such that self-compatibility is, or in the case that self-compatibility is more frequent on islands. So we set out to do this, and in part we did it in Solanaceae, which was why I'm showing you this result. So what you can see here on the right are the contingency tables for each of the three families. Here's the real focus of interest, and then also Brassicaceae and Asteraceae. And what happened is, in each case, we have the trend of a signal, an overall a strongly significant result, which explains about 10% of the variance for oceanic islands. In other words, breeding system explains about 10% of island mainland difference explains 10% of breeding system abundance of each state of or frequency of compatibles and incompatibles on oceanic islands. And this is, of course, somewhat erased by time, by errors in scoring, right, because of subsequent evolution, and by errors in scoring breeding system and by other potential processes. So it's a pretty interesting result, and also hints that there could be many other contexts, right, in which self-compatibility is favored. Okay, so there's some causes to believe that self-compatible mutants can experience advantages, and we in fact see this in high proportion of total species and angiosperms that are self-compatible. And so before we go anywhere else, what I'd like to do is return to something very fundamental, which are the genetic properties of self-incompatibility. We may need to understand these to both understand under which circumstances, for example, we can infer past losses of self-compatibility and unidirectional losses as well, so their directionality. Okay, so specifically for RNase-based gametophytic self-incompatibility, so this Solanaceae mechanism, the way it generally works is that if we take a style of a plant and apply, or a flower and apply pollen onto the stigma in the style, the pollen of the plant that we are applying pollen from, so its own pollen, is recognized and rejected. And this is really not a surprise to anyone. This is a relatively common phenomenon, as I mentioned before. So if you take a particular plant's pollen, it is rejected. Moreover, if you take the identical pollen-bearing identical alleles from different plants in this population, there also is the same response. It's as if it were self-pollen and no seed is produced. Now, you can find an individual in a population that shares just one of the two alleles in a genotype, such that, for example, the plant that was S1, S2, right, that's still our mom here, that's still the style, and the stigma is still the same genotype. But the pollen now is drawn from a different plant. Let's say there's one allele S3 in this genotype, S1, S3 of some other individual in the population. And what happens in this case is the S1 pollen is still rejected from this different plant, but S3 pollen is not. And so what will happen is it will grow, effect fertilization, and it will result in this segregation of genotypes in the seeds that are both now carrying the maternal alleles, S1 and S2, and then the paternal allele S3. And of course, it's entirely possible that in a larger population with working self-incompatibility system, all kinds of alleles occur. So alleles that don't match our mom, our S1, S2 genotype and in this flower. And so if we look at the stigma and the style of this particular individual, S1, S2, and place pollen that's from a different individual, say S3, S4 in this population, what you're going to get is both pollen grains will be able to grow. So you're going to get pollen tubes that effect fertilization, and then you get segregation of all these, Mendelian segregation of all these different S alleles. So there's an interesting prediction that this makes, which is that, first of all, in order for the system to function, you need to have at least 3 alleles. If everyone has the 2 same alleles, let's say in the best case scenario, they're heterozygous, they just have 2 alleles, no one can fertilize anyone else in such a population. So they need to have at least 3 alleles. Moreover, if they have a 4th allele introduced, you can see how that 4th allele may have some kind of an advantage, right? Because the other 3 now, S1, S2, and S3, are common in the population, say they're present about 1/3 frequencies, right? And so there are 3 genotypes, S1, S2, S1, S3, S2, S3. And this 4th allele, when it first comes into population, will be able to fertilize everyone. So it'll have an enormous male advantage, okay? So pollen side advantage. And so this is not a well-held secret. This has been known since, I think the first person to describe this was Sol Wright in 1939 to explain Emerson's data for Phaerothera. And so this process, or this mode of selection, is called negative frequency dependence. So the fitness of an allele depends on the frequency of the allele in the population. And so, for example, if we have our 3 alleles here, they'll be equally frequent at isoplethy, right, over some amount of time, if those are the three alleles that were present in this population. If the population is very large, they'll be present very similar or equal frequencies. And when we introduce this 4th allele that's initially extremely rare, right, it's going to have a huge advantage. And so in the next generation, what will happen is it will have such enormous advantage that it may in fact overshoot the equilibrium and then eventually settle at 1/4 frequencies, because we have 4 alleles. So the new isoplethy, the new stable equilibrium, is at or around 1/4. And if we introduce a 5th allele, the same thing will happen. It will have a very large advantage initially, except the advantage is attenuated. Now it's a little bit lower than the 4th allele had, right? And as we add new alleles, the 6th allele and the 7th allele and so on, they have smaller and smaller advantages. So that if you introduce, the 20th or 21st allele, they have about a 10-11% advantage. Still, I mean, it's an enormous amount of advantage. So this makes some predictions. One of the predictions is that there should be highly polymorphic loci governing self-incompatibility, okay? Another one is that alleles should be very difficult to lose out of these populations. All right, so We did some of this work in Solanum chilense, where we cloned and sequenced, at this point, some of this is unpublished, about 150 plants, and found, I think now actually 36 alleles. So the species-wide allele estimate is about 35 to 42 alleles, so matching Wright's prediction. And what's interesting is that pairwise amino acid identities are as low as 30%. What I have for you here is actually an excerpt of an alignment of such sRNase alleles from this wild Chilean tomato. And these are alleles. These are not different genes. They're alleles that occupy the same spot, right? They're opposite each other in the genotype, and they're immensely, immensely diverged, okay? So the reason why this happens is because negative frequency dependence results in this long-term maintenance of alleles in populations, and it results in another amazing phenomenon that we're going to end up using a lot later on for our macroevolutionary inference, or to sort of solidify our macroevolutionary inference. So here's the prediction it makes. If you have a particular species tree, so this is the sort of taxonomic set of systematic relationships among species, let's call them A, B, C, D, and F. And let's say that some of these are self-incompatible. So species A, C, and E are self-incompatible. What's going to happen is if you sequence their alleles, just like Solanum chilense that I just showed you, what we'll recover is this pattern of transspecific polymorphism, such that alleles from, say, species C and E are sometimes more closely related than they are to other alleles from their own species. So C to another C, or E to another E. And this is this pattern of transspecific polymorphism. So the polymorphism at the locus is very old. And so these alleles, because they're older than the species, the pre-existing polymorphism at the base of this species tree is passed on to each one of the species. And so you recover this incomplete lineage sorting that's really extreme in this case. And so in Solanaceae, here's what that looks like. So this is just a sample of all the alleles that are now available in Solanaceae, which is really, we have a spectacular data set at this point, submitted by many, many labs. So on the left, I have sort of a sketch of the species phylogeny for the species that are depicted in, for some of the species that are depicted in the tree. And really the take-home message here is that you have, for example, you have 2 species of tomato. We have a Lycium, Nicotiana, 2 species, and then we have a Petunia, okay? And if you look at the red color, the red solid color, that's Solanum chilense. It's this wild Chilean tomato that Iris Peralta actually mentioned, I believe the last talk. That species is present all over this gene tree. You can see all these red dots. And in many cases, it's more closely related. Some of these lineages are more closely related to Petunia lineages than they are to other wild tomato lineages. And in some cases, you can see, for example, and it didn't show it here, but in open white, sort of white and red open circles are Solanum pervianums. So they're very closely related to Solanum chilense, and you can see that they're paired up very often. And it's because we have very good samples from both of the species, and they are very closely related. So in that case, we recover this incredibly well-sampled transspecific polymorphism. Okay, so the sort of key take-home message from this is that the polymorphism of many S alleles presently segregating in self-incompatible species in Solanaceae was present at the origin, at least at the crown group of the species that I'm depicting here. And the take-home message from that is that if the transspecific polymorphism is present for all these species, say A, C, and E, right, for the incompatible species in the family, the key implication is that the common ancestor of all these species was self-incompatible, and moreover, these species A, C, and E are self-incompatible, and that self-incompatibility has been inherited by those species in an unbroken manner from the start of the family, so from the crown group all the way to today. And that's a really spectacular thing to be able to infer from gene trees and species trees. Okay, so what I'd like to do here now is take a look at the data from Solanaceae. So about 45% of the species are self-incompatible, so slightly less than half of the species are self-incompatible. And the rest of them are mostly self-compatible. There are only a few dioecious species in the family. What we did is we had this aging alignment that we made, and phylogeny that I'm not going to dwell on very much because there are many others that were subsequently made are far better than this one. But nevertheless, a pretty good sort of hypothesis for relationships within the family. We overlaid on this data on self-incompatibility. And so what happens is we got this fairly large data set of self-incompatible and self-compatible species, and using this sort of genius inference method called BISI that was described in this Madison et al. Paper by Saliato and colleagues. What we did is we inferred the rate of speciation and extinction associated with compatibility and incompatibility. And the sort of result here, which is at this point kind of old and not particularly interesting, is that self-incompatible species have relatively high speciation rates and much higher speciation rates than extinction rates. And yet self-compatible species have these much higher speciation rates and amazingly much higher inferred extinction rates as well. So if you look at their net diversification rates, if you subtract speciation so extinction from speciation, what we find is that the net rate at which they're diversifying is much higher for self-incompatible species. So the balance of births and deaths of species associated with self-incompatibility is inferred to be much higher than that for self-compatible species, which, by the way, is also negative here. So far, so good. Another inference that we made a couple of years later when Emma was here in the lab. By the way, Emma is now at Los Alamos National Lab, so it's very sad for all of us. But when Emma was in the lab, we were working on some model development, and one of the things that we worked out was a model in which allows the inference of cladogenetic versus anagenetic transitions in characters. And so with anagenetic shifts, what you have is a shift from incompatibility to compatibility. So it's depicted very simply here. So an anagenetic shift is in a lineage. There's some, over time, trait change from, say, self-incompatibility, compatibility, or some size metric, or any trait really. Whereas cladogenetic shifts are shifts that are associated with speciation events. So at speciation, there's a trait change. And so for us, a cladogenetic shift would be a shift, for example, where self-incompatibility transitions to both incompatibility and compatibility in such a way that at a speciation event, it gives rise to a compatible clade, as well as a self-incompatible descendant. And what we did is we tried to estimate the relative rate of both cladogenetic and anagenetic shifts, and determined that about the frequency of cladogenetic shift is about 10 times greater. So that's kind of an interesting result, and a result that makes some other predictions about sort of molecular signatures, at least at the S locus, of how this might work in self-compatible species, and so on. I won't dwell on it very much here. It's described in this paper in 2012. And then more recently, we also did some work that I'll sort of explain in a second, or why we were sort of insecure and why we were redoing some of this. So Rosanna Znoll-Ferguson took lead on this amazing project in which what we did is we applied this Boolean-Amara method called HISI, which allows a hidden state to be modeled along with the observed state. And in that way, you can think of it as some other state, or really an agglomeration of all other states. So self-incompatibility isn't the only thing to determine diversification dynamics, and this allows for some explanation of some slop sort of in that process. And regardless, we still kind of recover pretty much the same result, which is that the presence of self-incompatibility has a really strong signature on diversification dynamics. So breeding systems seem to be important in determining net diversification rates. So here for both hidden state A and B, so regardless of the background, self-incompatible species seem to still have higher speciation minus extinction rates. Okay, so in summary, the long-term diversification dynamics of Solanaceae seem to balance the common lower-level selection favoring losses of self-incompatibility, and most losses are associated with cladogenetic events. Now, these methods are not without problems. That's true for most inference, especially for this kind of soft inference where we have trees and states, right? And so there's been a large amount of work pointing to some of the sort of soft spots that we have in inference and really blind spots. And so one of the recent ones is this paper by Luca and Pennell, in which they show that there's an infinite really number of models with different diversification dynamics, but equal likelihood for any tree. They don't do it for these state-dependent diversification models, but it's very clear that the similar principles apply. And then there's a series of other papers, including these sort of main two by Rybowski and Emma, by Dan and Emma, and Wayne Madison and Rich Fitzgerald, in which they basically point out something. The sort of key take-home message here is that if you have a tree with very few transitions, you can get all kinds of artifacts and inflated type 1 or type 2 errors. So the real solutions for this are perhaps lacking, but we have some that are pretty good. So the one thing that we can do is expand the models to improve the approaches. There are some fairly clear ways forward on this. Nothing is really going to be a foolproof fix, but we can also examine some very awesome things that are associated, some particular traits that SI has itself that can help us with this. One of them is that the effect of SI can be examined as a replicated event on a wide variety of backgrounds of traits. And there are two reasons for this. One, as I mentioned before, there are all these different SI mechanisms. And then the second thing is that there are all these losses of self-incompatibility. So it's not just that there's one loss and you're looking at one clade that's SI and one clade that's SC. Instead, there are, dozens, if not sort of thousands of losses in the family over time. And so what's cool is that this allows us to perform replicated analyses, sort of both replicated within the family and also replicated in other families. And work in this vein is ongoing in the lab. The other thing that is kind of critical here is that long persistence of ancestral polymorphism in genes involved in SI allows us to essentially infer with a very strong prior belief that ancestrally in this family, the ancestral state was self-incompatibility, and that it has been transmitted to currently living extant species without any breaks. And so this is a really kind of powerful part of this inference framework. And this particular sort of thinking about this framework may not be unique to self-incompatibility. For example, others can use inferences from various sort of molecular details of their, or physiological or other chemical details of their systems. One example, for example, there's one approach which Stacy has been applying in her lab, where something about the chemical structure branching and the genes involved in creating those, and we can know a little bit more other than just having tips and trees to perform inference of trait evolution. All right, so in this vein, what we decided to do is expand our inference to other families. So what we did is we went on a big fishing expedition across eudicots. The general approach is sort of described here, which is that we would like to find something, so ideally genes. This is a candidate-based approach, so we know that we're looking for these T2S-type ribonucleases. They're in a particular sort of group of ribonucleases. We know that they should be expressed in the style. In the pollen, we should have these other genes that are called S-link F boxes that I haven't really mentioned very much. They are sort of the male part genes in this response. We're looking for self-incompatible eudicots because that's who should express these genes, perhaps. We're looking for, as I mentioned before, at least 2 alleles per individuals, and also per each individual, and also a highly polymorphic locus. So a locus that should have many alleles segregating the species, and also have this ancestral polymorphism. So the alleles should be highly divergent from each other. And moreover, they should have the correct phylogenetic placement. In other words, these alleles that we find should be in this ribonuclease family. And we tried dozens of species, but here what I'm going to show you is just results for a plant that's very dear to me, which is Schlumbergera. So this used to be called epiphyllum. It's about to be a plant that's about to be everywhere in your grocery stores and windows and so on. Right around October is when the sales start, and they start flowering around Thanksgiving, Christmas, roughly in the winter. So we're going to move this work to Cactaceae. We've sampled saguaro and some other species, where about 65% of the species are self-incompatible. So slightly more species are self-incompatible than they are in Solanaceae. There are about 1,600 species in the family, and they're a little younger than Solanaceae, although who knows. So this is how our sRNase fishing expedition proceeded. We had some previous data showing that Schlumberger was gametophysically self-incompatible. This is from Thomas Boyle at University of Massachusetts. So what we did is for style expression, we sequenced using RNA-seq styles from 13 individuals. We got a whole bunch of reads. We performed de novo assembly. I'm not going to bother you with the details here, but what we did is we assembled reads from pooled RNA-seq for each individual. They're barcoded so we could separate them. And we assembled a joint transcriptome of about 40,000 genes, sort of in quotes. So these are just gene assemblies with about 60,000 isoforms. And our candidate, an RNase that seems to be exactly right, was one of the top expressed genes in the style. So that's good news. And then we examined pollen expression, same thing. We found some candidate genes, and I'll show you that in the following slides. So what we did here is for each accession, which will be the rows in this particular depiction, in this illustration. So each plant, each accession is going to be on the left. And then for each row for that plant, I'm going to show you the expression data. It'll be just false color expression data for RNases, so the style part component or pistil part component, and for pollen part components on the right. All right, so here are our accession names or numbers. And for each accession, what you'll see is the expression of two different alleles. So for example, HO1, HO2 share the genotype AB, 3 is BE, and so on. Each one of them expresses two stylar alleles. And then a number of F boxes, which is consistent with how, I've sort of papered over this, but it's consistent with how the Solanaceae mechanism works. So far, so good. It looks like we have great data from Cactaceae, and it looks like in Schlumberger, at least, we found about 5 alleles. So this is a cultivar that's, bottlenecked, so it's not a surprise that we find few alleles. And 5 genotypes. That's not necessarily true, by the way. It would be just 5 alleles and 5 genotypes. It could have been a different number of genotypes. We also found that pollen expression data confirmed presence of these pollen part genes. And if we make a gene genealogy or a gene phylogeny for this particular type of RNase from Cactaceae, what we find is that from Schlumberger, they're highly distant or distinct, just like they are in Solanaceae. So their polymorphism seems to be ancient. Moreover, we had the good luck of sequencing 2 rare species, or a single individual, and find 2 alleles in a rare species of Matucana from Peru. And it looks like just from those 2 alleles that there's transgeneric or transspecific polymorphism in Cactaceae as well. So we have stylar expression of the correct gene, pollen expression of the correct gene, in other words, the same gene family as in Solanaceae. We, I'm not showing this, have crossing that's consistent with single locus gametophytic system, phylogenetic analyses that place these as related to other RNases in this particular group. And we have transspecific polymorphism in Matucana and Schlumberger. So it looks like Cactaceae expressed the Solanaceae mechanism. And so the next question that we are sort of interested about, and obviously, is to replicate our data so that we can have sort of a greater weight of evidence supporting the previous findings of Solanaceae to sort of buffer against these criticisms of methodological flaws, right? And so what we'd like to do is replicate the net diversification rate analyses in Cactaceae. And this is work that's actually ongoing right now. And I'm just going to show you some preliminary data. So remember, in Solanaceae, we found the net diversification rates were higher for self-incompatible species than self-compatible species. So using a series of very sort of similar approaches, we found that, we made a large phylogeny. we had about 750 species of self-incompatibility and compatibility data, so the breeding system data, which is quite a bit, actually. It's kind of surprising. So we combine those, and we find in this sort of highly preliminary analysis is that in Cactaceae as well, we seem to be able to replicate this result. So the net diversification rate of incompatibles there as well is higher than it is of self-compatible species. So this is a fairly exciting finding for us. We're actually working on some other families as well, but the real sort of take-home message of all of this is that it seems like we have a quite a bit of weight of evidence at this point that the RNase-based incompatibility, which was found both by us and by other labs across all of these different families, or here orders of pentapetalae, seem to indicate that that group, as an ancestral state, had self-incompatibility. We don't have transfamilial or whatever transorder polymorphism. The polymorphism at the locus is confined to families, but it's interesting that this particular mechanism is so widespread. It's found in so many different families across this tree of angiosperms. And it looks like pentapetalae is really where it originated. There's a number of reasons why I think this is true, but I'll omit them here. I'll just point you to Carlos's paper from 2017, which is fantastic. All right, so in summary, self-incompatibility is present in a number of distantly related families, and it's caused by this homologous mechanism, the Solanaceae type mechanism. The mechanism is likely ancestral for this very large group of angiosperms, so in pentapetalae. Our approach, this brute force approach for using RNA-seq, seems to really open the door for a lot of work on difficult non-model species. So, you know, if you guys think that there's a new self-incompatibility mechanism somewhere, this is one way to actually also start discovery of a new self-incompatibility mechanism. We used it just alongside this sort of candidate gene approach. The macroevolutionary consequences of this form of self-incompatibility seem to be mirrored in Cactaceae, so we recover very similar results as in Solanaceae, and this is pretty exciting. And then this approach that we took, this RNA-seq based approach, which is a totally brute force, seems to really work well. And that's a little disappointing for me because I like finesse, but if brute force worked, that's what we're going to use. All right, well, with that, I would just like to thank the many people who've been working on the projects today, directly in the lab, I mean on the projects that I showed here. So that's Carlos Hermanauskas, pictured here on the bottom right. Lucy Delaney, who's also in my lab, who's been lately working on distribution of self-incompatibility in Fabaceae and Orchidaceae. Emma, who is sadly now, or I mean, good for all of us because she's at Los Alamos National Labs where she's working on COVID and many other things. Rosanna Zanil-Ferguson, who is at University of Hawaii, Manoa. Dina Grossenbacher, Matt Ritter, and Jen Yost at Cal Poly. The Baker's Law Group, and then these people at Huntington Botanical Gardens, including John Traeger, Dylan Hannon, and Jim Folsom, who were really helpful in our Cactaceae work, as well as Ken Olson and Joey Santore, who gave us samples of Schlumberger that proved to be kind of key, these unique genotypes, and then National Science Foundation for funding. And with that, I would love to open it to questions if anyone has any. I see that there's some chat messages. Perhaps those concern the talk. But for now, I'm just going to stop sharing. And
awesome. Thanks, Boris. So now we can open up for questions. So you can type in the chat or you can unmute your mic. Maybe Sandy, you want to start with your question?
Okay, just to say, I put a picture of Mike in the chat as well, a link to a picture of Mike if you want it, Boris. So, so I wanted to know from the Goldberg et al. Paper, you had a very high sampling in Solanum when you inferred the speciation and diversification rates. You had a really high sampling of the potatoes because the potatoes all had molecular sequence at that time. But other clades, which are equally, if not more species rich, weren't included in that tree. So could that result be biased by the fact that you had bias in the clade, in the species inclusion in different clades?
Yes, the rate inference could easily be biased by many, many things, right? But I think that so the rate precision will be affected by something like that very clearly.
Because when we
accuracy.
when we did a past history and included all of the species of Solanum, the highest diversification rates were in the arid zone of Australia.
Yeah, that's what we found, and that we found. I mean, we did that analysis on that tree originally for that paper, and we ran some of the.
No, but it's a different tree, so this is a different tree. We used
right. We found the same result, is what I'm saying. In our tree, is that the Australian Solanums have an immensely high diversification, right? But the thing there is that there's something called a sampling fraction, which is that we assume that we don't have a total sampling across the tree. It's also true that that sampling fraction applies to the whole tree, so that local disparities in sampling effort, so in how much we sampled for each group, may make some difference. But I will also say this: if we just say, okay, let's just say, is there a way? So if we apply something like a non-parametric method, so forget BISI for a second.
Yeah.
So Emma and I did this about 10 years ago. So if you say, okay, what if the transition rate from incompatibility to compatibility is some relatively high rate? And we estimate that from the tree. And then we say, is there any way after, say, 50 million years to see any SI species, any self-incompatible species, given that their rate of loss is really high, but the net diversification rates are equal? In other words, if SI and SE have equal net diversification rates, but SI is being lost at a high rate, as high as we observe, would you see any SI species at the end as a proportion of the total? And the answer is that you wouldn't see as many as we see today, not even close. In many cases, you would see zero,
that's right. Yeah,
right?
that's right.
And so the part that's robust is that the net diversification rate of incompatibles is very likely much higher than the net diversification rate of the compatibles. Now, the specific details, where on the tree the rates may be shifting, and the rate associated, say a compatible species goes to a new continent, right? And there it diversifies much more than it did on the let's call it mainland, right? And Australia is enormous, but say we're talking about Australia. Clearly there you have another trait that's affecting diversification. That's not just SI, it's not just breeding system, right? And so, yeah, that would affect the sort of the rate for that particular group would be a little bit different than a rate for some other group. But
I mean, across all of Solanum, the diversification rate, the net diversification rate in the self-compatible Australian Solanums is higher than anywhere else on the tree, including in the potatoes, which are all self-incompatible. But what's interesting about our results as compared to yours is that we also get higher than background diversification rates in the potatoes, which are self-incompatible, but not as high as in the spiny Solanums, which are self-compatible. And that's with the all species tree. That's all species included.
Right. And one possibility, right, Sandy, is that but say you're ignoring the fact that self-compatibles may go extinct rapidly, right? So there's a lot of pool of the recent on the Australian.
Yeah. No, that's true.
Right. So if they have a high turnover, which is what we're predicting, right, they may boom and bust over time. We don't know that, of course, right? This is a prediction that we'll have to be here in 20 million years to see, but it's possible.
Yes, yeah. Well, we can talk about this later, but yeah. Anyway, I just wondered.
Awesome. So we also have a question from Stacy that I will read out loud. She has two questions. So what is the fraction of SISC and Cactaceae, and how does it compare to Solanaceae? And then she has a second part of the question I'll read right now. So what factors determine the depth of trans-taxon polymorphism, and why does cross-genera not say families? What decays this signal? Periodic bottom.
Yeah.
Chelsea?
Yep.
That second question, I'm going to ask you to read in a second again, but let me just answer the first part. The first part was the fraction of SI species in Cactaceae and Solanaceae.
Yeah, and
so the fraction of self-incompatible species in Solanaceae, to the best of our knowledge, is about 45%, plus or minus a few percent. The proportion of self-incompatible species in Cactaceae is about 65%. plus or minus a few percent, right, from our sample. And now the second part of the question.
Why do you think there's a difference? Do you think it has to do with lifestyle, or just if you had to speculate?
If I'm invited to speculate, I will.
Yes.
So, as I often do. So there are all these associations between breeding system and some other traits. One of them is that, for example, the habit. So trees or tall statured, large statured things tend to be self-incompatible more so than annuals that are small, right? Or woody species, non-woody species, same thing. So in cacti, that's a little bit complicated because the description the words that we have for those don't translate very well to, say, Solanaceae, right? Globose, right? Or that doesn't really have an equivalent in Solanaceae. But I would say that they tend to be perennials and more long-lived perennials. And they have that phi ratio, the ratio of mitoses to meiosis. That is, or meiosis, that's much, potentially much higher. And so that's one reason why they may have a greater proportion of self-incompatibility. It could be ecological too, like it could be purely by something not, like they are not found on islands very commonly or something like that.
Yeah.
Right? We'll find out. Well,
Yeah.
we'll look for it anyway. Dina actually is working on this right now. Dina Grossenbacher and I have this collaboration. We're trying to work on some of this stuff.
Awesome. Okay, and then the second one, what factors determine the depth of trans-taxon polymorphism? Like why across genera, but not, say, across family? And then what's the decay signal? And then periodic bottlenecking, how does that play into this?
Excellent questions. I papered over all of that. So we think that there are these bottlenecks in some species. I think is the last reference, or some, maybe not species, some genera or some anyway groups that reduce polymorphism ancestrally. So this would be in Physalis and with Thuringia. What we think determines the age of polymorphism is the strength of negative frequency dependent selection and population sizes, so effective population size, and mutation rates also possibly. So what would happen is that if you have very, very large population sizes, you can maintain many of these alleles, and if those population sizes are stable over time, you can maintain very many alleles for a very long time. We don't have a particularly good model linking age and turnover of alleles. There's some attempts at this by sort of Adam Richman and Mikkel Schurup and some people in Denmark. But it's not clear to me why there are no transfamiliar polymorphisms, nor that they don't exist, right? We're just talking about unfathomably huge amounts of time. And so imagine finding, like incomplete lineage. So imagine how hard the job of a systematist would be, or a phylogenetic systematist, if any gene behaved like self-incompatibility genes, right? Where you can't tell family, like you have trouble telling how families are related, right? I don't know exactly why we don't see transfamiliar polymorphisms, but certainly population size scaling is very important in determining the age of polymorphism.
All right, cool. Thank you. And then MSU Potato, do you want to ask your question? You can unmute your mic. If not, okay. So his question, or their question is, did you find Cactaceae non-sRNase factors such as HT that would be involved in SI responses? We can't, you're muted, Boris.
Sorry, now I'm not.
Yeah.
Sorry, I lost everything for a second. I unplugged something by accident. So we have not yet in detail looked for other factors. We will do so soon. We just have this sort of pile of sequences, and it's hard enough to make solid inferences on sort of the basic portions of what we have. And we concentrated on the things that are really they're not modifiers of the response or are necessary for the response, but we concentrate on things that are actually interacting to determine allele recognition identity for now. But that's a great idea, of course, and it's something that can be also done in other families, right? And also in places where when you lose self-compatibility, when some group loses self-compatibility, for example, you have entire families that don't have any incompatibility, right? So within Solanaceae, there's some big groups that don't have any incompatibility. And what we could do is try to examine how incompatibility was lost, whether it was lost multiple times. And so these auxiliary factors, including RNases, F boxes, and then HT and all of these other genes could be used to figure out what was broken, when it was broken, which groups share monophyletically or sort of as a synapomorphy, loss of incompatibility, and so on. And so they're really important for this kind of inference.
Is that things that did you look at that when you looked at all of Solanaceae, of like looking at the sRNases, but also like these HT proteins and other things, these other proteins that are involved? Do you find that there's a large variation within that family, or not something you looked at?
Large variation in?
Like the HT expression in colon 1, those kind of other proteins that are involved in self-incompatibility.
We haven't done any of that work. So that work is done. There's a group that's been working, sort of a large group from Colorado State and Missouri and Indiana. And they've been working sort of on unilateral incongruity and expression of all of these factors, additional genes. There are many, many genes required sort of for proper function of incompatibility. It makes it kind of like an eyeball, right? Like if you break this thing, it's very unlikely that it will re-evolve easily, especially once you collapse the polymorphism at these RNase and F-box loci. It makes it very hard to regain with the same genetic machinery.
A little bit different next time. The next question is Gustavo. Sylvia, do you want to ask your question?
Gustavo,
yes.
go. Oh.
Thank you.
Yeah.
Thank you, Boris, for your talk. I am just curious about the source of the new alleles of these the S alleles. Do you think that it comes just by mutation, or there could be another forces shaping the new alleles, the source of like recombination, structural variants? And at this point, it could be important to understand the change from the self-incompatibility to compatibility, and also some important thing in the colonization process. As you say, it's most easy to colonize if you are self-compatible. And then maybe you can get the compatibility or evolve the compatibility, and maybe it could come after, for example, after divergence migration from another species for a sister species. So what could be this the source, or what do you know about this, the source of the S alleles, the new alleles?
So I think there may be two questions there. Was one about how new functional S alleles within self-incompatibility are formed, and then the other one, how breakdowns work, right? Like whether it's mutation or recombination, or is it just allele identity that you're asking about?
Yeah, mainly the source of the functional alleles to make the self-incompatibility.
A lot of people are interested in that question. I'm least likely to find the answer to that question. So but again, I'm invited to speculate, so I'll just go ahead. Alex Harkness, and who's a shared student between Emma Goldberg and Yaniv Brandwein, has been working on just a theoretical model of how it is that the male and the female portion, so pistil and pollen part, co-evolve, right? Because they have to keep in sync. If you recombine the male and the female portion of the response, what happens is you get a compatible individual. And so it's kind of interesting that therefore, at least in tomato and in some other species, the locus is the male and the female component are very closely linked. And they're also centromeric. So there's this like remarkable region of suppression recombination found right in that locus. And the locus is huge. A centimorgan in this locus is about maybe 20 megabases. or maybe more. Someone should look at the exact data, but it's enormous. The size of the locus is huge. The physical sort of nucleotide size of the recombination distance is enormous. So it looks like that doesn't suppress, gene duplication still. So paralogs, it looks like these F boxes are paralogous copies of themselves, right? So there may be still some gene conversion. There may be rare recombination that's contributing. But the thing that we can observe most easily is just mutation, right? Like point mutations, indels, and things like that. And they are abundant. If you take two S alleles, we don't have very many, but if you take two sequenced regions and you align them, there's just like rearrangements and massive deletions and all kinds of retro elements and stuff. It's really immensely complicated. And so one of our goals is actually to sequence a couple of these haplotypes we have already sequenced from cultivated tomato, and now we can sequence them, some closely related alleles in Solanaceae, which is sort of the focus of some of the research in the lab. And the idea is to get at your answer a little bit. So we can then see, right, what exactly is going on in multiple copies of the same allele that may be in different populations, maybe in different species, but the same allele, and then also closely related alleles, and then compare the structure. And that hopefully will let us maybe begin to answer your question.
Yeah, that's cool. Yeah, because you mentioned a rate about the 10 to the power -5, something like that. So I guess mutation is not enough to explain this fast rate. Maybe there is another factors involved there. And just one little thing, always these genotypes will be heterozygous in self-incompatible species, right? There is no point, there is no way to be homozygous. So, for example, if I am looking for some possible individuals that lose the self-incompatibility, the first thing is try to find like some possible homozygous individuals in the allele side. For example, in some edge of the distribution of the species or something like that. I guess that could be the main way to start looking for specific individuals or population that lose the self-incompatibility, right?
So I'll start with that last part of the question. So nearly every self-incompatible species that I know of anyway that's ever been examined in any detail contains self-compatible mutants, so individuals that are segregating the population, generally recent ones, that are self-fertile. So the rate of mutation seems to not be limiting how frequently compatible individuals arise. And then the first part of your question was, you said 10 to the -5, 10 to the -7. That's the rate of recovery of self-compatible pollen grains from self-incompatible individuals. Mm-hmm. And the key thing there that you're suspecting is too high to be point mutation is polyploidy. So polyploidization has a very high rate, and it obligately in Solanaceae breaks down self-incompatibility. And so I think that's what's causing the really high rate very, very often. And that may be in part the reason behind that high cladogenetic shift estimate, right? Is that when you have polyploidy, you also have breakdown of incompatibility. And so now you have this like vastly different taxon that's split off from your original taxon. At least it's very highly likely to be described that way. So taxonomy and taxonomic description has an enormous role in what we do. And I think actually to Sandy's point, one of the things that's kind of interesting is that species descriptions in different places in the world can play a huge role in determining the precision of the rates that we're inferring. So that too is a big sort of interactor. And it's not a trait, right? It's just taxonomic practice. And then the last thing you said, it was about homozygote formation. So you can make homozygotes sometimes with bud pollinations and with some treatments. And in Petunia, people have been particularly successful at doing this. We've never succeeded in Solanaceae. I've done just about everything. And we got some seed that were selfed from an incompatible individual. And we grew them out. We got 4 seeds after like thousands of crosses, you know. And we grew them out, and like I think 3 or 2 of them were achlorophylltic. They were just like whitish yellow mutants that obviously had some problems. So inbreeding depression is really, really high. And that makes it a little bit difficult to work with these generated homozygotes.
Okay, great. Thank you very much.
All right, maybe we can squeeze one more question in before we have to go. So there's Federico.
Yeah, Federico has a question.
Yeah. Do you want to add disease? Yep, there we go.
Yeah, hi. So Boris, I think you have already kind of answered, I happened to work with a plant which was kind of messy in terms of self-incompatibility, which was flux. And it had some characteristics that I didn't know if they are common. And I wonder how common they are, even in the Solanaceae, and if that will mess a little bit about the theory about how self-incompatibility evolves. And how common is it to have species that have both self-incompatible and self-compatible? I think you already said that is kind of common. The other is that self-incompatibility doesn't segregate as a binary trait, but as a continuous trait. And the final is species that have both gametophytic and sporophytic systems. Do you know if that's common? Because sometimes, sometimes people describe a species as something, but they don't really test it in a way that will allow us to know these things. Do you know if those things are common, or it was just that I was unlucky to work with that plant?
So, when Mendel started working on his experiments, the first thing he did was he worked on peas. And then I forget, well, it was like, I forget what the second species that he worked on was, but it was like apomictic.
Hyraceum. It was Hyraceum.
There you go. And it was a real mess, right? It was like he got all kinds of results that he couldn't really follow up. Like there's nothing was clear. Nothing was segregating him into the ratios, right? And I feel like, this happens. I think the reason why a lot of people believe that self-incompatibility is just like not at all like a qualitative trait is because in some systems, especially where you have breakdown of incompatibility, so you have either segregating incompatibility and compatibility together with many mutants possibly, right, segregating, so the independent mutants. When you do the crosses, you're going to recover some very strange things, right? We can predict that's in fact expected. And so just to show you something, I think I can still share my screen. At the back of my presentation, let me go here and see whether I still have this. So we try to examine this phenomenon in some detail, and here it is. Okay, so we try to examine this phenomenon in some detail, and what happened is we looked at the distribution of the index of self-incompatibility. So what happens if you take in a large population or species, whatever, if you have some samples, you self-fertilize them and you outcross them. And then you score an index of self-incompatibility, which is 1 minus the relative success following selfing divided by relative success following outcrossing. Okay, so if it's at 1.0, that means that there was no seed set after selfing and some seed was set after outcrossing. And if it's at 0 or less, the plant set as much seed following self-fertilization as following outcrossing or more, right? And so if you do this for about 1,300 species of angiosperms, here's the distribution that you get. And there's a lot of stuff in the middle, right? So these here, crossed out, hatched out marks are negative, so they're effectively 0, right? They're sort of normally distributed around 0. And then there's some stuff in the middle, and then there's some stuff that's pretty high. And I think, Federico, what you're doing is working somewhere over here on the right side. And so if you look at, you see these notches below? So those are individual species that we have data for many, many individuals within the species. And so they involve these species here. Here's our Solanum chilense. It's self-incompatible, and it has some individuals that are self-fertile, right? So we uncovered these in the course of doing hundreds of crosses in hundreds of plants in the greenhouse from wild populations. And then if you see something like, this prunus, it looks like it has a whole bunch of segregating self-compatible mutants. But then if you look at the Spartina, I don't know what that is. That may not be incompatibility at all. So it's really difficult to tell sometimes what's going on. But sometimes, even though it's easy, the system is relatively clean, you get these in-between species-wide estimates of ISI, and that could be true for populations. I don't know. I don't know if your guys have this. Let me quit this. So I think that partly might explain what's going on, as well as just like you could have a ton of inbreeding depression, right? So you can have loci that have very high inbreeding depression that are linked to your failure of seed set. And also some kind of mechanism, like a real SI mechanism. And that would really make the data very dirty. And I'm sorry, by the way.
Yeah. I just hope that it's not that we are biased by the simplicity of the system that has been very well explored in Solanum. And it's very simple, and it has to be simple to work, actually. But maybe it's not that simple genetically in all plants, and we just haven't explored enough. Maybe.
No, it's clearly true in some groups. I think in your group, it's definitely true. I think across Fabaceae, it's going to be true. I think like in large swaths of, maybe even in Solanum in Australia, I mean, there's some that are supposed to be self-incompatible. We'll see.
Okay, thanks a lot.
Oh, thank you. It's good to see you. It's good to see you again. And I'm sorry it didn't work out. We tried RNA-seq and your stuff, and nothing there.
I think Iris has a question. I missed her question earlier, so if she wants to ask.
Please go ahead.
Hello, Boris. Great talk. Now, my question was about what species Solanum abrocaites and Solanum penelli. Those are self-incompatible. But at the borders of their distribution, they became self-compatible. And I wonder if you find something similar in Solanum chilense. And the second part of the question, if those populations that are at the border of the distribution, the appearance of self-compatible could be driven by pollinators, by the lack of pollinators, for example. It's interesting because in Solanum arcanum, a northern species, they have several self-compatible populations, but not in the other self-incompatible species. So what do you think?
So I think, so in Chilense, we found a smattering of an individual here and there that's self-compatible. But then we also found it's not really at the traditional range edge necessarily. It's sort of like in the, not range edge classically, but it's a population that's sort of distinct from other populations in that it's isolated, which is common for many populations of Solanum chilense. That's sort of a coastal Pacific population in the south. And it seems to have a higher frequency of self-compatible individuals, and may be in fact self-compatible entirely. It could be that it's just the apparent self-incompatibility could be caused by inbreeding depression, by just early acting inbreeding depression, which is very high in the species. So we're not sure which of those it is, but we definitely uncovered some individuals that are totally self-compatible, where we get enormous seed set following self-fertilization. And of course, as you know, this is a widespread species. I'm sure there must be a population. Like I would bet money on there being a population that is entirely or almost entirely self-compatible somewhere. In fact, even many populations near each other or far, right? And I think what drives that probably, I think you might be right. I think that absence of pollinators in some of these populations are probably the drivers of the advantage that the self-compatible mutants have. In Chilense and in other species, as you mentioned.
Have you studied any of the Solanum arcanum?
We have some sequences from Arcanum, but I haven't done any sort of field work on them or anything like that. I only have TGRC accessions, right, for those.
Because to me it seems to be the opposite than Chilense, that probably in Solanum arcanum group, like Chimeluski and Neoriki, they are self-compatible. And also they are closely related to the Lycopersicum group. Those are all self-compatible. So, I mean, it will be interesting to contrast as different lineages where incompatibility and self-compatibility probably have to do with the lineage diversity and adaptation to different ecological needs, something like that. I don't know if I can explain, but it's a very interesting model to contrast Chilense, your Chilense with.
Yeah, so I think
it will be,
I think you're right. I think there are some really serious differences. I think Chilense has much larger effective population sizes, for example, than the entire red-fruited group probably combined. And then, or whatever, some polymorphism. I don't. It depends on how you measure polymorphism or diversity. But one thing that's interesting is that we and some other groups have been sequencing S alleles from the red-fruited species, and they will definitely not have anywhere close to the diversity of alleles that a wild species might have, like Chilense. I think that there are about 2 distinct sequences that we've recovered from Lycopersicum. One is very similar to a sequence in Pimpinellifolium, but they're all very closely related to each other. It looks like it's possible there was a single breakdown of incompatibility, and then there's only one or maybe a couple of very closely related alleles from that group that are now inherited by all of the red-fruited species. And that's a sort of an interesting question, right? How many alleles survive the breakdown, and when they become neutral, right? So eventually they'll all be pretty much gone, right? They'll coalesce very, very quickly.
And if you add the tomatoes from Galapagos, could be also that they inherited that sequences as well. I mean, the Chismani and Galapagos.
Yeah, that should be, of course, they should have even lower diversity no matter what, right? So you're exactly right.
Well, thank you very much.
Thank
Good
you
luck.
so much. Yeah, thank you. I will email you right after this is done.
Okay, I have some ideas that we can share.
Lovely, thank you so much.
And one more thing. Well, we have been also studying with other students the Portulaceae. You know that they are divided in many, many families right now. But the interesting thing is that they are very, very well adapted to very hard extreme environments in the mountains, in the Andes. And we studied some of the populations, and they are all self-compatibles. Self-compatibles. And those families are at the base of the cactaceae. So this could be also interesting for you to explore the Portulaceae. It's a basal lineage.
There are many, many lineages between the origin of Pentapetalae and the currently extant species that have lost incompatibility and no longer express. There are no individuals that have any incompatibility. All of Apiaceae. I'm not aware of a single report of incompatibility in Apiaceae, for example.
No, in Portulaceae.
No, I understand. I understand. But what I'm saying is that in other families too, right? There are many, many incompatible families, and around them, there are plenty that don't have any, that are sort of at the base of some group, right? And I'm glad to hear that it's also in Portulaceae, or whatever sensu, the new Portulaceae, right?
Okay. Thank you.
Thank you so much.
Awesome. Thanks again, Boris, for giving a talk and sticking around for the discussion. And then I'll see everyone next week. All right.
Thank you.
Okay.
Thanks,
Thank
Sandy.
you, everyone. And remember, next week we have the talk of Gustavo Silva about wild tomatoes.
I think the ecology of wild tomatoes, right?
Yes, sorry about that. Yes, wild tomato ecological genomics, elucidating adaptation process to stressful environments. So see you next Friday.
Bye, Boris.